This podcast will be an overview of the cell cycle, and then we'll do a brief podcast on mitosis and another brief podcast on meiosis. Whenever I talk about the cell cycle, I like to start it with this very simple slide and just ask the question, what is 1 to the 46th power and what is 2 to the 23rd power? I ask this question because it's a good, simple way to understand the difference between mitosis and meiosis. Clearly 1 to the 46th power is 1, and when you think about mitosis, the object of mitosis is to produce genetically identical daughter cells from one parent cell. On the other hand, if you look at 2 to the 23rd, that's a really big number. It's more than 8 million, and that's a good way to describe meiosis, because in meiosis the chromosomes behave as pairs and they segregate independently of each other at the first meiotic division. We have 23 pairs of homologous chromosomes, so at the first meiotic division there's a minimal potential of more than 8 million genetically distinct types of cells that will be going into forming the gametes. Your learning objectives for these 3 podcasts: you should be able to describe the events that occur in each phase of the cell cycle. You should be able to describe stages of mitosis and meiosis. It's useful to be able to do this by comparing and contrasting. You should be able to describe the concepts of C and N, and you should be able to describe how cyclin and cyclin-dependent kinases regulate passage through the cell cycle. The central events of the cell cycle would be duplication of chromosomes, separation of chromosomes, and cell division, or formation of daughter cells. We like to say that these stages occur in discrete, non-overlapping stages. The cell cycle is progressive and it's unidirectional. That means it only goes in one direction. You have to have DNA synthesis that occurs in the S phase of the cell cycle, and then chromosome separation occurs in the M phase of the cell cycle. And for mitosis, there's 1 round of DNA synthesis, 1 chromosome separation. Meiosis has 1 round of DNA synthesis and 2 chromosome separations. We've come to understand over recent years that heterodimeric proteins control the cell cycle. There's a regulatory cyclin subunit and a catalytic cyclin-dependent kinase subunit. And this cartoon is just meant to give you that view. So there's the regulatory cyclin subunit and the catalytic cyclin-dependent kinase subunit. And as we've said, the cell cycle is an orderly sequence of events in which a cell is going to reproduce by duplicating its contents. That's going to occur in interphase, and then it's going to divide, and that's going to occur in the M phase of the cell cycle. We understand that there are 3 components to interphase. There's a G1 component, an S component, and a G2 component. Then there are 2 components to M phase. There's karyokinesis and cytokinesis. Now let me just quickly review these. S is the synthesis phase. G1 stands for a gap phase before DNA begins to synthesize itself. So it's a gap from the end of cell division to the start of DNA synthesis. G2 is a gap from the end of DNA synthesis to the start of cell division. And then we talk, of course, about a G0 phase where cells have differentiated and have taken themselves out of the cell cycle. Cells in very early embryos don't have significant or protracted gap phases, so the cell cycles in early embryos pretty well progress rapidly through DNA synthesis and cell division. We've come to understand that there are several cyclin-dependent kinases that regulate passage through the cell cycle in all cells, and this would include human cells. And this slide is a little bit of an oversimplification, but it has the main points that I would want you to worry about for our course. we can talk about a G1 cyclin-dependent kinase complex. The cyclin would be cyclin D, and that causes cells to pass a restriction point. And you might think of this, if you want, as the start of the cell cycle. It's a little bit artificial, but it helps to sometimes think about things in a linear manner. So we could say the G1 cyclin-dependent kinase complex allows cells to start the cell cycle. Then we have an S-phase cyclin-dependent kinase complex. The main cyclin here would be cyclin E, and that's involved in initiating DNA replication. And then there's an M-phase, or a mitotic cyclin-dependent kinase. The main cyclin here would be cyclin B, or for historical reasons MPF, for maturation phase promoting factor. And the M-phase cyclin-dependent kinase complex allows the cells to progress through mitosis, or there are mitotic cyclins that allow the cell cycle to progress through meiosis. This is a little bit more complicated view of the cell cycle. This table and figure come from the textbook that we use for the course. It's beyond the scope of what I want you to know for our course, but it makes a couple of interesting points. First of all, the figure shows you in general where the cyclin and cyclin-dependent kinases are active in the cell cycle, tracing through from G1 through S, G2, and M. And then the table shows you some of the targeted phases of the cell cycle where these cyclins and cyclin-dependent kinases operate, and gives you an idea in broad scope of some of the effector proteins that are targeted. And notice that some of them are tumor suppressor proteins, others are proteins that regulate passage through the cell cycle, various transcription factors, things like nucleolamins might be affected, etc. This is way beyond the scope of what you need to know for our course, but I put it here just to give you a general perspective of what might be going on in regulating the cell cycle. Going back to a simplified view of things, we can say that activation of cyclin-dependent kinases would require cyclin binding. So here's a mitotic cyclin-dependent kinase binding onto the mitotic cyclin. That activates this cyclin-dependent kinase. That would, for example, trigger machinery for mitosis. Likewise, to inactivate the cyclin-dependent kinases, you have to degrade the cyclin. And the only way you can go from one stage to the next in the cell cycle is to degrade the appropriate cyclin to progress forward. So in this simplified figure, it shows the activation and degradation of the mitotic cyclin, or on the bottom, the activation of the S-phase cyclin-dependent kinase, and then its subsequent degradation to progress from G1 beyond S into G2. This basic understanding of the cell cycle led to the Nobel Prize in Physiology and Medicine. This shows a simplified human somatic cell cycle. Across the top, one can see the stages of mitosis. So there's prophase, prometaphase, metaphase, anaphase A, anaphase B, and telophase. And then the simplified diagram shows the major stages of the cell cycle. Intraphase, consisting of G1S, G2, and then mitosis. And then, of course, it shows differentiated cells, cells that go into G sub 0. And for our course, we might think of differentiated cells as cells like neurons or cardiac myocytes, any cells essentially that no longer divide. Some cells remain in a prolonged G1 stage. So, for example, adult stem cells for various organs or tissues within organs might be in a prolonged G1 stage. Memory cells of the immune system would be in a prolonged G1 stage. And we've already told you that in embryos, G1 and G2 are very brief. In a typical cell cycle, G1 is usually the longest phase. G1 and G2 can be variable, but G1 is usually the longest phase in a typical somatic cell cycle. This again is a simplified overview of cell cycle time, and we sometimes in our mind's eye think of the cell cycle as a 24-hour time frame. That may or may not be accurate, but we can just kind of make a point in rapidly dividing adult cells. Most time is spent in intraphase. So for example, through G1, S, and G2. And rapidly dividing cells like the bone marrow, cells in the GI epithelium, or tumor cells, spend most of their time in intraphase. The bottom piece of the cartoon is just to remind you of the importance of the cytoskeleton in relation to the cell cycle. Clearly, the spindle fibers for moving the chromosomes and the contractile ring for separating the cell during cell division. The spindle fibers, of course, are mostly microtubules. The contractile ring, mostly actin interacting with myosin. Now, somatic cells can be classified by their mitotic activity, and we talk about static cells. Those would be cells that are no longer dividing. They'd be post-mitotic cells. They're usually long-lived cells. So, for example, cells of the central nervous system, skeletal muscle cells, cardiac muscle cells, we usually consider those as static cells. We can also talk about stable cells. These are cells that might divide episodically to maintain tissue or organ structure. They can be stimulated to divide more rapidly if there's an injury. So, for example, we might think of periosteal or perichondrial cells as stable cells. Smooth muscle cells would be stable cells. Endothelial cells, fibroblasts, stable cells. These would divide episodically, but if there's an injury, they can divide much more rapidly to heal the injury. And we can talk about renewing cells. These would be cells that display regular mitotic activity, and the daughter cells will either differentiate or they'll remain as stem cells. Differentiated cells may be lost from the body. We can talk about a slowly renewing population of cells. They may increase in size. So, for example, smooth muscle of hollow organs, fibroblasts in the uterine wall, lens epithelial cells, or even slow-growing tumor cells. These will be slowly renewing cells. We might also talk about a rapidly renewing population of cells, like the precursors of the blood cells in bone marrow, epithelial cells and dermal fibroblasts of skin, the mucosa lining of the GI tract, or tumor cells in rapidly growing tumors. These would be rapidly renewing cell populations. Here is a simplified way to look at the cell cycle. We talk about G1, we talk about S, we talk about G2. Remember that in S, DNA replicates. So in the simple-minded diagram, we could say this cell would be 2N and 2C. This is in G1. As the cell passes through S, the DNA is replicating, and as the cell is in G2, we would say that that cell is 2N4C, and then as the cell goes through mitosis, for example, two daughter cells are produced that are back to 2N and 2C. So here I want to define for you the concept of N and C, first in general terms and then specifically for humans. When we talk about N, we're talking about the haploid number of chromosomes for a given species. N is unique to that species. For humans, the haploid number of chromosomes is 23. C is the amount of DNA in a haploid cell. Usually we take that as the amount of DNA in a mature sperm. So in humans, C is 3.5 picograms of DNA. Here's an interesting diagram that looks at DNA content in human somatic cells, looking at the DNA content by a fluorescence-activated cell sorter, or a flow cytometer. On the y-axis, one plots the number of cells. On the x-axis, one plots the relative amount of DNA per cell. And you can immediately see that there are three populations of cells in these types of experiments. There's a population of cells that have not replicated their DNA. That's what's shown by the cells that are in peak A. These would be cells in G1. And then there are cells that have replicated the DNA. These would be the cells represented in peak B. And those could be cells that are either in G2 or in the early M phase of the cell cycle. And then there are cells that are intermediate between these two peaks. These would be cells in the S phase of the cell cycle. And you can notice on the x-axis that there's a relative increase in DNA amount as you move along the x-axis. Another point to make from this type of experiment, if you look at the distribution of cells, there are more cells in G1, and that's how we've come to understand that G1 is the longest phase in the cell cycle. Now, I told you that the DNA content for human sperm equals 3.5 picograms. With that information, what would the DNA content be for the cells that are represented in peaks A and peaks B? I hope you'd understand that peak A would represent cells that are 2C before DNA replication. They would have 7 picograms of DNA. Cells at peak B would be in G2. They would have a 4C amount of DNA or 14 picograms of DNA. Here I'd like to spend just a few minutes and talk about cell cycle control. And the first point I want to make is that cell cycle control relates to the fact that the stages of the cell cycle occur in a sequential or progressive manner. This is a cartoon that comes out of the Alberts textbook, and I really like it because it's so oversimplified that it makes good sense. This is a washing machine dial. And if you think about the way a washing machine works, to wash your clothes, for example, before your clothes wash, this machine has to fill with water. Once the machine fills with water, the machine washes the clothes. Once the machine washes the clothes, the machine empties the water and then rinses the clothes. You can't go backwards. You can't rinse the clothes before you wash the clothes. So it's a simple-minded analogy, but it's a good way to explain how the cell cycle works. It occurs in stages, in sequential stages, and it occurs in a progressive manner. And to move from one stage to the next requires that certain events must occur, just like in the washing machine. So with respect to the cell cycle, you enter the cell cycle, you trigger DNA replication machinery. Only when that machinery has been triggered can DNA be replicated. You cannot trigger the mitosis machinery until DNA is fully replicated. Once DNA is fully replicated, the machinery to trigger mitosis is activated, and then once that happens, the mitotic spindle is assembled. Once all of that occurs, you can complete cell division. So again, sequential, progressive manner. This allows the cell to go through various checkpoints. So for example, you can say, is DNA replicated? The G1 checkpoint would say, is DNA replicated? If it's not, then you stop, you replicate DNA, or you repair DNA if it's damaged, and then replicate DNA. Are the chromosomes on the spindle? If they're not on the spindle, you stop and attach the chromosomes to the spindle. That's necessary before the cell can divide. So the checkpoint signals act as brakes or negative controls to halt or slow progression through the cell cycle. Checkpoints allow input of extracellular signals from neighboring cells as well. So we can say that control of the cell cycle is all about triggers and checkpoints. This is a very much more complicated slide than we need to worry about for our course, but you can see some major triggers in the cell cycle. There's an S phase entry point, there's an M phase entry point, and then there's an anaphase and telophase trigger point. And then you can see a variety of checkpoints around the cell cycle. Again, more complicated than we need to worry about, but there's a unreplicated DNA checkpoint, a spindle assembly checkpoint, a chromosome segregation checkpoint, and then look at the DNA damage checkpoints that can operate all the way around all the triggers in the cell cycle. So again, a much more complex slide than we need to worry about for our course, but you can ask the simple question, why do you have to have cell cycle checkpoints? That would be an absolute requirement to prevent catastrophic genetic damage, and this simple-minded cartoon shows what happens if there's a failure of the chromosome segregation checkpoint. You would get non-disjunction. Somehow the chromosome segregation checkpoint fails. Not all the chromosomes are attached to the spindle, so when the cell divides, one cell is going to miss a chromosome, whereas another cell is going to get an extra chromosome. You can imagine that could lead to very catastrophic genetic consequences to the daughter cells.